Hi guys, I hope you're all doing well in this quarantine situation that we are still finding ourselves in. And I still remain hopeful that we will be going back to school soon. I don't know anything, but I'm sure we'll all find out generally around the same time what's going to be happening for the next upcoming weeks. So I'm going to try to make the most of this time, and I hope you guys do too. If you have any questions on the assignment for the week, please reach out to me. I sent you a long email with a whole bunch of information in it. Everything is accessible on Canvas, but at the very bottom of the email, there is a little table that tells you when the best time, there are 2 times per day, except for Friday, that you'll be able to reach me most easily. If you need to send me something outside of that time, please, of course, do. I definitely will get back to you. Just make sure that you give me a little bit of time in order to do that, because we do have meetings scheduled and like department meetings, faculty meetings, and other things that we might be doing at other moments during the day. So this week is about restriction enzymes, and you guys have a little bit of background with that because you've been doing some assignments that have to do with CRISPR. And CRISPR is a restriction enzyme. It's a restriction endonuclease. So nucleases are enzymes, ASE, and what they do is that they cut genetic material. They cut nucleic acids. So we're going to talk about restriction enzymes in general because they've existed long before our discovery of CRISPR, and they are all pretty cool, and we've been using them for a very long time in the field of biotechnology in order to do lots and lots and lots of different things. So I know there's a lot on the screen right now. We're not going to go through all of this. We're only going to go through just the part about restriction enzymes, but I will kind of maybe come back to this particular Prezi, which I did not make. I borrowed from someone because it's really good, and I didn't want to reinvent the wheel. So there are some big questions. We're not going to go through all of them right now, and we will kind of address all of these in the next upcoming weeks as we move through our curriculum. But, there's relationships here we have to explore. There's moral and ethical and legal obligations that we have to, as scientists or consumers of science, be ready to kind of grapple with. So all of those are kind of overarching questions as we move through biotechnology. And we are going to start here with restriction enzymes. So how do they work, what they are, and then some examples of a couple. Okay, so remember, please take notes as you go. So restriction enzymes, how they work. Hopefully we know restriction enzymes are naturally found in bacteria. So that's where CRISPR is found, and again, it is a restriction enzyme. And what they do is they are DNA cutters because bacteria are constantly under attack from things called bacteriophages, which are viruses specific to bacteria. And as we know, viruses inject genetic information, and they hijack cells in order to use the cell's machinery to make more viruses. And that usually kills the cell. So restriction enzymes are kind of basic, and I hate the word primitive, but I'm going to use it here, primitive immune system. So our immune system is a lot more complicated, but bacteria needed, in their world, they needed a way to protect themselves. And so the bacteria that had the ability to make restriction enzymes had a selective advantage over ones that did not, because they weren't actively getting killed by these bacteriophages. So restriction enzymes are just enzymes. They are proteins that fold into a specific shape. Remember, shape is important. And they're made by the cells that they work in. So a particular cell makes a restriction enzyme. Not all bacterial cells make all restriction enzymes. Some are specific only to certain kinds of bacteria because they get made in response to infection, to exposure to foreign or viral DNA. So it just depends on maybe like what they've been exposed to. So what they do is they scan DNA. They're constantly kind of scanning around, grabbing onto the DNA, unwinding it, and looking for their sequence. And what they recognize is something called a palindrome. So think of like the name Hannah, Hannah. Hannah is the same forwards and backwards. And that's what these particular enzymes recognize. So in our 4 letters of DNA, they recognize sequences that are the same forwards and backwards. And they can do one of two things. They are going to cut the DNA. When they see their sequence, every single time they see their sequence, they're going to cut it. And they're going to cut it either in blunt cuts, which if you watch my mouse right here, imagine that the DNA, we know it's, sorry, my cat is hitting my screen. She loves the computer. Blunt cuts, if you watch my mouse, it literally would just cut straight down. Okay, so through both strands, kind of just like a knife through butter. Those are blunt end cuts because they kind of just end. So both parts just are done. But a lot of times they will produce something called a sticky end, which is this. So in a sticky end, the cut is not straight through. It kind of cuts one backbone and then cuts the hydrogen bonds and then cuts through another backbone at a different point, through the other backbone at a different point. And it creates these overhangs. So you have this sticky end here and this sticky end right here. The reason they are called sticky ends is because these nucleotides, whatever ones are kind of left hanging out on the edges, they want to make those hydrogen bonds. That's what's holding the two strands together naturally, is the hydrogen bonds that exist between nucleotides. So they want to make hydrogen bonds, so they're kind of primed or ready for making connections with pieces of DNA, not necessarily this piece over here. But that's why scientists have used them, like to use them, because we can do things in this space. We can insert new DNA. We can kind of look at putting something else in there and what might happen if we do. So sticky ends for our purposes are going to be more common because they're a little bit easier to work with in the terms of what we would want to do in biotechnology. Okay, so what are they? Restriction endonucleases. So it's not going to zoom in, but look right over here. That's where we're going right now. So restriction endonucleases, again, endonuclease just is the name of the enzyme. They cut only at a specific sequence. Okay, so it's a restriction site. They're usually between 4 and 8 base pairs long. They're not going to be bigger because it's harder for then those sequences to exist. Like, it's less likely that a longer sequence will exist, but a shorter sequence, you have a kind of a higher probability that you're going to kind of see it and recognize it. And they're going to cut through the DNA. So whether they cut blunt ends or sticky ends, they're still cutting the DNA. And many have been isolated from all different kinds of bacteria. So this is very common in the world of bacteria that these restriction enzymes are made. And again, like I said, they serve as a bacterial immune system against phages, bacteriophages, which are the viral attackers for DNA, or sorry, excuse me, I'm having a stroke, for bacteria. Here's a picture of a really common one. So this right here, this guy right here is EcoR1, and you see the molecule of DNA right here. So the green and the kind of pinkish purple is the DNA. The EcoR1 is the restriction enzyme. It wraps around the DNA. And it's going to scan along. So it's going to scan, kind of open up the DNA and see the sequence. It's going to read the sequence. And then once it hits this particular sequence, GAATTC, remember that's one strand. So the other strand would be GAATTC on this way. And it's going to make the cut. And right here, you can see the cut. And that's a sticky end cut. So you see how there is like an overhang right here and an overhang right here. And every time this particular restriction enzyme sees this sequence, it's going to cut. And what you might be wondering is, well, how the heck does the bacteria prevent this particular enzyme from cutting its own DNA? Because I bet you this sequence, GAATTC, exists in the bacterial chromosome. And that would be really bad if this enzyme cut it. So it goes to DNA packing. So what the bacteria do on their own chromosome, where they see this sequence, they actually add a methyl group, which, if you remember, winds the DNA up really tight so that the actual restriction enzyme can't bind and can't cut the DNA at that point. So that's kind of how it protects itself from the restriction enzyme. It methylates the point on its own DNA where the restriction enzyme would want to cut, so it can't do it. let me go back for a second. Restriction enzymes are named for the bacteria that they come from. So EcoR1 comes from E. coli, and it was the first one that was isolated. There are EcoR2s, EcoR3s, but this was the first one, and it's a pretty common one. You'll see this often. And they're all recognized different restriction sites. So this one is PVU1, and PVU1 comes from a bacteria called Proteus vulgaris, which lives generally in your intestine. It can cause infections. It's not super common, but its restriction enzyme is going to be recognize a different restriction site than EcoR1. So if you see right here, we have our cut here. So this is a sticky end cut. And we have our A and T and T and A, so it was cut kind of between there. So if you look, it's going to recognize this sequence. It's going to cut up, if you watch my mouse, here, here, and here, okay? Right there. It's a really short cut. I mean, it's about 4 nucleotides, but that's what it does. So that's every time it sees that, it's going to cut. And the cool thing about restriction enzymes is they allow us, again, to combine DNA, make recombinant DNA, and we can insert DNA from organisms into other organisms using this. And you kind of see it in this picture over to the side right here. So we have our segment of DNA. We have a restriction site. This is for EcoR1, GAATTC. It's going to make the cut. We have our sticky ends, and then we have this gene of interest from another organism, denoted in gray. And because we have sticky ends that were cut by the same enzyme, so if you look at the sequence here and here, they would match, and here, they would match. So it makes it easy to insert that gene right in here. And what's kind of cool about this and why we reviewed is that we need an enzyme we are already familiar with to come in. So you see these breaks in the backbone. That is where our friend DNA ligase, which remember we talked about with DNA replication, is going to come in and seal up. It's like our glue, our molecular glue. It's going to seal up those ends in the backbone and make a full recombinant DNA molecule. So when you hear recombinant DNA, you need to think DNA with genes kind of from different places. And oftentimes we use it to make something like this, which is a plasmid. And we'll talk about plasmids next week or the week after. But they're a tool that we use in biotech very, very, very often, often in medicine, to make products that we would then need to give to people. So we'll talk about all of that. But that's where we are going to stop for today. So it's a pretty short set of notes. Again, I hope you're all doing well. And please, please, please reach out to me if you have any questions about what's the plan for the rest of the week.